Hello everyone, my name is Hannah Kim, and today I'll be sharing my research on developing new biomarkers to improve immunotherapy response in lung cancer patients. Lung cancer is the leading cause of cancer-related deaths worldwide, and also one of the most commonly diagnosed, accounting for roughly 1 in 8 new cancer cases per year. A promising type of cancer treatment is called immunotherapy, and it works by activating the patient's own immune system to recognize and attack cancer cells. And these therapies can involve using immune cells, antibodies, or even vaccines designed to help the body fight cancer more effectively. While immunotherapy can be life-saving for some patients, it only works well in about 25 to 30% of lung cancer cases due to several reasons. Some tumors have genetic mutations that make them resistant to immune attack. The tumor microenvironment, surrounding cells and molecules, can be hostile and block immune responses. And finally, each patient's baseline immune status differs, meaning that their body may respond differently to treatment. And this is where my research comes in. My goal is to develop methods to identify biological markers, or biomarkers, that can help predict which patients are more likely to respond well to this immunotherapy treatment. A biomarker is something measurable in the body that tells us about a person's health or disease status. In cancer, biomarkers are often proteins that are found on cancer cells, and a special group of these are called glycoproteins, which are proteins that have carbohydrate or sugar modifications on their surface. Abnormal patterns of glycosylation can indicate the presence of cancer cells, and so because of this, glycoproteins, or glycobiomarkers, can be especially valuable in understanding treatment responses in lung cancer. My research question was, how can we better detect and analyze glycoproteins to identify new biomarkers that can predict immunotherapy response? To answer this question, I worked on developing methods to both visualize and analyze these glycoproteins in lung cancer patient samples. First, I worked with lung tumor tissue samples by preparing very thin sections of tissue and staining them using immunohistochemistry, or IHC. This method uses antibodies conjugated with a color-changing enzyme called HRP to reveal the presence of CD8 positive immune cells within the tissue. And these are immune cells that directly target and kill cancer cells. Interestingly, in patients who responded better to immunotherapy, we saw clusters of these CD8 positive cells gathered around the cancer cells, suggesting that the immune system was more actively engaging with the tumor. Next, I used lectin staining to detect glycoproteins. I compared results with and without a reagent called p-engase-F, which helps release sugar groups called glycans from proteins for easier detection. You can see in the p-engase-F treated group, the green FITC dye was a lot dimmer compared to the control, meaning that more glycans were released from the proteins, indicating that glycans can be successfully separated for further analysis. To go beyond the tissue level, I also isolated proteins from peripheral blood mononuclear cells from patient blood samples, immune cells, and analyzed them using mass spectrometry, a technique that identifies proteins based on their molecular weight and structure. I used bioinformatics tools using pGlyco software to focus in on glycoproteins that were detected. In total, I identified around 190 potential glycoproteins. And interestingly, most of these did not have common sugar modifications like sialic acid or fucose, which might suggest unique glycosylation patterns in cancer patients that could be linked to differences in their treatment response. Overall, my results show that glycoproteins can be successfully detected in lung cancer tissue using staining methods. And I also identified glycoproteins present in patient blood samples that could serve as potential biomarkers for predicting immunotherapy outcomes. These findings lay the groundwork for future directions. The identified glycoproteins can be further screened at the individual protein level, lay a preclinical foundation for future biomarker discovery, and advance precision medicine and improve diagnostic accuracy and patient outcomes in lung cancer. These are my references. And I'd like to thank the Zhang Lab at Shanghai Jiao Tong University for providing me with the opportunity to carry out this research, as well as the U of C Study Abroad Office for their support. Thank you so much for listening.